Yes, I'm here.
Junmin is an accomplished researcher and expert in the field of proteomics and neuroscience at St. Jude Children's Research Hospital. Over his career, Dr. Peng has published in excess of 240 manuscripts and has been invited to present at numerous conferences worldwide. Included in his research is the use of human disease models to study protein turnover and drive insights into protein dynamics and potential therapeutic strategies. We are very pleased to have Dr. Peng speak today on the methods and biological significance of some of his proteomics work.
Right.
Floor is yours.
Yeah, I would like to thank for the invitation and then for this opportunity to share some of our work. And then is the slide shows up well?
Yes.
Okay. So yeah, and the title of my talk is Exploring Proteome Turnover in the Marine Model of Alzheimer's Disease. So actually, I'm actually at St. Jude Children's Research Hospital. Many people ask me where it is. Actually, we are located in Memphis. And Memphis is a city named after the ancient city Memphis in Egypt, and which close to Nile River. We have a Mississippi River right here, so you can see the Rainbow Bridge right here, and then our hospital is right here, and we also has a pyramid as well in Memphis. So my lab is largely focused on proteomics and metabolomics technology development, and also application in different biological fields. So you can see here, we actually develop a methodology, and then also largely one of my main interests is to analyze the protein ubiquitination and protein degradation. So my interest in protein degradation has been rooted a long time ago. One was a postdoc in Dr. Steve Gige's group at Harvard. And then at that time, we know the protein can be ubiquitinated by a E1, E2, E3 enzyme to form either monoubiquitination form, and then which can fully extended to polyubiquitinated form through lysine-48 chain or through lysine-63 chain. So if they go through lysine-48 chain, that labeled protein will go to proteasome, get degraded. And then if you label it by the other, maybe they will go to non-proteasomal pathways. So during that time, actually we invented a method. If we trypsinize the ubiquitinated species, the ubiquitin is cleaved at the arginine right here, and then to generate a small glycine tag. This is a signature GG tag. And then this tag add a mass addition and 114 to modified lysine. And then also this modified lysine, it will not recognized by trypsin anymore, and then showing as miscleavage sites. So using these approaches, we began to profile ubiquitinated proteome, and then in numerous papers later, and then we found actually interestingly, ubiquitin can form chains through all the lysine on the surfaces, not only lysine-48 and then lysine-63. They can also through lysine-6, lysine-11, 27, 29, 33, and then also even N-terminal. So actually this opened up a whole new field called ubiquitin code. And then now actually has been well received in the field. People recognize ubiquitin can form many different chains. So like a tree can grow linear or they grow branched, right? So through all different conjugation sites. And then many of them involved in proteasome degradation, but some of them actually involved in non-proteasomal pathways. So in addition to that, the specific GG tag has been used to generate antibodies. And then using the antibodies, and then this specific GG tagged peptide can be immune affinity purified and to analyze ubiquitinated protein. At the same time, we all know actually you can analyze whole proteome as well. So with the two-dimensional proteomics data, whole proteome and ubiquitinated proteome, we clearly would like to understand how protein get degraded, right? What's the degradation rate? So for a long time, actually, it turns out to be quite difficult to do it in animal. Although the method has been very well developed in cell culture model a long time ago, you can see here, this is a review paper to show if you have this cell culture using the Select method, and then you can change it to the heavy medium. And then during this process, you can see some protein get labeled, right? And then in this way, and then you can begin to use that to measure the decay of the unlabeled protein to measure the protein degradation rate or even synthesis rate. So, but however, you could do the same thing in animal, right? You can see here, and then you can use this lysine diet, and then we actually obtain from Cambridge Isotope. Right? You can see during a time course, you can observe the protein get labeled with time, and then appears to be good to analyze. But however, there's a fundamental differences between these two experiments. If you look at the left side, you can exchange in the medium almost instantly, right? It's very easy to do. You just switch in the medium, right? So, and then you can, in this way, you can largely replace the lysine and free lysine, light lysine with heavy lysine in a very fast rate. But however, you cannot do this in animal, right? In animal, the free lysine can be generated from two sources. One is from the food, another one is from internal degraded light proteins. So that actually the lysine itself has a kinetics. So then you can see here, if you get two protein degradation rate from the cell culture, right? And in the animal, you can see the decay of these protein A, protein 1, protein 2 in the animal is largely affected by the unbound lysine pools, because the lysine pool cannot be switched immediately, right, in animal models. So this is just extending this slightly further. You can see how this works. So if you simply fit the protein P1 and P2, and then without consider lysine curve, you get a half-life like 18 days and 13 days, right? So this is a regular fitting, right? You get the half-life. But however, if you consider the delay of lysine, what you really should observe is the distance between the protein curve with the lysine curve. So the half-life is much, much shorter, right? It's only 7 days and 3.5 days. You can see the difference is very large, right? So in order to do that well, we actually spent a significant effort to generate a model use ordinary differential equations. So you can see here, the food has been taken by the animal, right? Get into the cell, and some of those get degraded, lysine get degraded, and then some actually incorporated into the protein. At the same time, the protein can degrade it in the lysosome, inside of the cell, or by proteasome. And then those lysine will be reused, right? Recycled inside of the cell. So this is some of the ODE equation we're using, so then we're using those full animal modeling. So we began to actually get a more accurate half-life analysis. And then in these cases, you can see that's the half-life we observed initially in about 3,000 proteins. So you can see even the neuron is not dividing. The half-life, the medium half-life is about 4 to 5 days. And then some protein actually turn over really fast, right? Less than 1 day or 1.8 days. Some is a medium, right? Actually, some of them has a relatively long turnover rate. So you can see here, like Dr. Jiang has initially published, some protein have a much, very long half-life. And then here we found the long half-life protein are generally in mitochondria and in mining sheet and also the integral components of the membrane. So now once we have the technology, we would like to apply those technology to study Alzheimer's disease. You know, Alzheimer's disease is the most common form of dementia. It's actually the top 10 leading cause of death in United States, and then it's a very costly disease. And then even in the last year, the cost for the disease is almost like $300 billion. And then now it's about 6.5 million people in US is affected by Alzheimer's disease. But this is a very large number. Oh, excuse me. Compared to about 20 million actually cancer patients in United States. So in Alzheimer's disease, and then you can see protein aggregation, right? Like the tau and A beta. And then also the protein aggregation events also occur in other neurodegenerative disease, such as prion disease, polyQ, Parkinson, and then other Pick's disease. So in Alzheimer's disease, we actually initially studied many neurodegenerative human samples directly from control patients versus Alzheimer's disease in the different human cohorts. So you can see one data set from Mount Sinai, New York. Another data set is from Banassan in Arizona. So each dots represent a single protein. You can see we found two pattern of proteins. One pattern is the protein accumulated in AD, right? You can see in both cohorts. And then the second pattern is protein decreased in AD, right here. If we compare the protein with an RNA data on the right-hand side, it's quite interesting. About 50% of the protein we see dramatically increased in AD is not changing at RNA level. So the shield is black. So that indicate the RNA level is not a good prediction to the protein levels. So now we are wondering is whether the protein degradation play an important role in this process. So using this animal model, and then we began to actually label the animal at a different date, like from 0, 4 days, 8 days, 16, and 32 days. So theoretically, you will see the light protein will be gradually replaced by heavy proteins, right? And then if you see the decay of the light protein, you will see a diagram like this. So we did a very deep analysis, use a TMT-based mass spec analysis. We analyzed almost 8,000 proteins in the mouse brain. And now we actually fit all those data in the same shot, use the differential equation, the model, and then we can begin to see those kind of fitted line. And then based on that, we can derive the half-lives for every individual proteins. So just here, quickly show you a very, very simple example. If you look at the protein APP, right, A beta and APP, A beta has been known to be the most aggregated peptide in human patients, also in this animal model. So it's a very short sequences, right? It's like 42 amino acid long, and then sometimes it's 40. So you can see they can generate this very specific human A beta peptide after trypsin digestion. And then at the same time, the full-length protein is present as well in the model. And then the full-length protein here, and then if you compare this human version with mouse version, because the mouse have their own APP protein, we have this specific human peptide. So this allows us to analyze human APP and human A beta degradation rate based on those two specific peptides. So the result is quite interesting. If you look at a human APP, this degradation like curve is like here. And then if you look at the free lysine curve, and then it looks like this. So if you put them together, you suddenly recognize the human APP degradation curve and the free lysine curve are very close to each other. Based on our modeling, this shows very, very fast degradation rates. So basically the degradation can occur within one single day. However, if you look at a peptide, right here, the A beta peptide, and then the A beta peptide put together with this full-length protein is dramatically different. So the A beta degradation rate calculated in the model is almost 40 days. So that kind of explain why the protein doesn't change that much. But however, in our brain, this peptide, once they generated, they aggregated, they have a much, much slower degradation rate. They began to accumulate it in our brain. So at the same time, we actually find other protein also actually shows much faster, much slower turnover rate, and it shows a higher half-life, right? So including ApoE and the CAR216, and then there's another protein called VTN and also NAMP2. You can clearly see the degradation curve actually gets delayed in the F80 model, which is the 80 model for the Alzheimer's disease. So finally, we actually, based on the data, we proposed a model, right? So in this model, we believe that A beta is the original components form those aggregation in our brain. And then once they form this aggregation, they actually generated a toxic microenvironment, which can trap many other proteins in that specific area. And then those proteins potentially also shows slower degradation. And then once you have a slower degradation, you get accumulated. So those proteins will play a very active role in this microenvironment to generate, to lead to toxicity. So we already know in the synapse and many other cells activated around the plaque area in the human cells. So I think this approach, turnover approach, allows us to the first time to monitor this degradation really inside of those brains. So yeah, at the end, I would like to thank the people in the lab to work on this project. And here is our current lab. And then we actually have people working on the mass spec approaches, work on disease animal models. And then we also have a Center for Neurodegenerative Diseases provider services. And then I also would like to thank the NIH and the internal support, and then many people provide us human samples for these studies. And also I would like to thank the organizer for this invitation, and I will be glad to take any questions.
Thank you, Junmin, for that excellent talk. We have time for a question or two. So I'll start off with one on my own. What approaches can be taken to integrate protein turnover data with other types of omics data?
Yeah, that's actually a fantastic question, right? So now actually with the proteomics tools, you can get many type of data, like whole proteome, right? Ubiquitinated proteome, proteome turnover. So actually this model, the current method can be used to study any phenomenon which potentially perturbed at the protein turnover rate. For example, like a PROTAC molecular glue, now it's very hard in pharmaceutical field, right? To actually selectively target some protein degradation. So what we would like to say, if the protein is actually perturbed, you will see the ratio between the ubiquitinated form versus the whole form changes. So that means the percentage of the ubiquitinated form will change. And this can be, if they are increasing, this may be correlated with the fast turnover. If they are decreasing, this will be correlated with a slower turnover. So I think that's one for the easy way we can think about it. Yeah.
Thank you for that thorough answer. We do have a few questions from the audience. We have time for one or two before moving on. What would be a future study based on the results presented?
Yeah, I think this is the beginning of the whole method, because you can see the method was just published only last 1 or 2 years ago. And we actually spent a significant effort to figure out how to do this differential equation model. So I think that turns out to be actually difficult, but finally done. So I think now it's possible to use this method to analyze protein degradation in real animal, in many different animal models, not limited to Alzheimer's, right? I think protein turnover is one of the big regulation events. So you can imagine you can use for this for certain cancer model, other neurodegenerative models. In addition to that, it's also possible to study even local protein degradation. For example, you can analyze the plaque, analyze the protein degradation, analyze the surrounding area. In addition to that, you can also analyze the degradation rate in different subcellular location by fractionation, right? So this is all possible, right? Currently, we only analyze the whole tissues, right? I think those are all potential methods can be done in the future. Yeah.
Lots of different directions for
Yeah,
sure.
yeah.
One more question, and then we'll move on. What's the price of your 13C amino acid diet? And do you have a preferred company that you ordered the diet from?
Yeah, clearly, I think we get from Cambridge Isotope. I think Andrew, probably you're the best person to answer that question.
Yeah, we'll have to speak offline about that. Well, thank you once again, Junmin, for that excellent talk. That brings us to the conclusion of today's Mass Spec Isotope Day webinar. I'd like to thank all 5 speakers for their excellent talks and everyone in attendance for your time and attention. Circling back to the goals for today's event, this session provided you with a platform to learn from prominent scientists in the mass spec field who utilize stable isotope labeled standards to fulfill different application purposes. The hope was that you learned something new, strengthen your understanding of CIL's labeled products, and stayed engaged throughout. At the close of the Zoom session, you will be prompted to complete a short general survey. It would be highly appreciated if you could take a few minutes to complete that. You should also expect to see in the near future a follow-up email with links to today's event, as well as the relevant information. Should any questions or product requests arise, please feel free to email us or visit CIL website at isotope.com. Coming soon in the weeks ahead is the NMR and environmental sessions of Isotope Days 2023. Their formats will be constructed in a similar manner as today's Mass Spec Isotope Day. The NMR is next Thursday, September 28th at 10 a.m. Eastern. Then the environmental the week thereafter on Thursday, October 5th, also at 10 a.m. Eastern. We look forward to seeing you all there. Thank you once again, and have a nice rest of your day.